yeah, and so this big table here is going to be the, and that's what we're going to conclude with, is going to be the entry point for any biological statistical analysis you're going to do. And any issue, and what that also means is that any issues that occurred above this in the pipeline are going to be flowing into your statistical analysis. Okay, so is this clear? Any question up to here? So yeah, so these two steps here. Where my mouse? I have to point with the mouse, otherwise I can't see. I think on the internet. This step here, and then this step here. So this step here, getting the BIOM files from all of these 3 file types we worked on, and then this step here, just getting just counts from our alignments for each of our samples. So yeah, really the first thing we're going to start with is aligning reads to a reference genome. So we star the star aligner, which is one of the programs you can use for this purpose. And we're going to assume that the star genome index has been built previously. So I did that. I'm not going to ask you to do that because it's long, because it asks for a lot of computational resources. But that's one step you pretty well have to do. And basically, this is what I did. So it's, as I mentioned, it's already done. And then I just use this FASTA file containing the mouse genome assembly, the chromosomes that we went over yesterday, and then the GTF file with the genes, the gene positions. And then, so that's with star. And this genome index is going to be a format that's specific to star. You can't use it for anything else than run a star with it. And the first thing you have to do if you want to work with star on the cluster is load the module. Otherwise, it's going to tell you command not found. So don't forget to do it. And then, yeah, so the last part is just the command that I use for creating the database. So just so that you have it when you will need, if you want to use star, when you will need to recreate that. So this is a FASTA file downloaded from Ensemble, which is I've created a genome directory on the cluster in the Scratch directory for the workshop. So you can find it there, but you can also just go to Ensemble, go to the FTP, download it. And then this is a GTF file. And I just create a directory for the database. And then I run star in genome generate mode, which is create the genome index. And then I tell it where the FASTA file is, and then when the GTF file is, and where I want the database to be. And that's pretty much it. And all of this I did within a grid job. So we're going to have an activity today. For those of you who are more comfortable with shell, I think you can work on that. But eventually, like as you're going to be working more on the cluster, you're just going to learn how these batch jobs work. And yes, so this database creation, I just submitted it, this command here, just put it in a script, submitted it as a batch job, and then it took a few hours to run. I probably should mention I used eight processors here. I actually haven't put the option here, but you pretty want to use several processors. Should not make, it's just only should make a difference regarding how long the job takes. Yeah, and I use 92 gigabytes of RAM memory, which is quite a lot. You have to know that star is using a lot of memory. That's what I was mentioning, that you should ask for more memories for your interactive jobs for today. Okay, so this is for the database, and so we have the database. It's somewhere in the cluster. We're going to say that in a minute, and then this is really just to align reads, and then so this is going to be the first activity for today. And so we're going to the cluster. I'm not sure if you already all have interactive jobs. So you're going to load the star module. You can use star.h to see the different star options. There's a number of them. And then we're only going to work on one sample for these alignments. And then I'm going to show you a bit how you can extend it to just work on all samples. Okay, so we're just going to work on this P10 rep1 samples, and then again we're going to use the 1/10 files so that the runtime is not too long. And then so you give it, you store somewhere in the variable, or you could write it directly on the command line as well, but it makes things more structured. So you give it where the reads are, and then where you want the resulting alignment to be, and then you just create that directory, and then you also record somewhere where the star database is, and then you call star, telling where the database is and where the reads are and everything. And this should all work. And then one important parameters, maybe two important parameters. Oh yeah. So one. So maybe the most important parameter is this one. So the star has this weird naming option convention, where so this is a filter. So it has a bunch of different filters. You can I would even encourage you to just go through the help. It doesn't have a manual page. Everything is in the help, so the help is fairly long. That's why you want to use it less. And then so the out filter is going to change which reads are considered aligned. And then in this case, it's reads with a maximum number of 5 mismatches. Okay, and there are like a few parameters that you can play with this, but in this case, in general, aligning to the human or mouse genome, especially here, it's going to be the same mouse strain that the genome is made for. So it's going to be very close. We don't really expect any strong deviations. So 5 mismatches out of 76 bases, which is the length of our reads, is going to be plenty. And then we want to only the last parameter, the multi-map number max, is how many multi-mapping reads you allow. So some reads might, depending on the sequence, map in different places in the genome. So in that case, we're just going to exclude all the reads. And finally, what we want is for the output type, the output format, we want an output in BAM format, which is the standard output format, and then we want the BAM to be sorted by coordinate. I think the default for star is outputting SAM, and it's pretty historical, but we want BAM and then sorted by coordinate, which is really the normal thing. Okay, so that's it for star alignments. So you should be able to get this these alignments to run for this one sample, and I think I've created a second, or you could try on like all samples and kind of get an ID. Yeah, so I want everyone to work on this, and there is more later. If you're done with this, I'm going to give you some time for everyone to go through this, and then we're going to directly switch. It's going to take some time to run, and we're going to directly let it run and just directly continue with counting alignments from files that are already created. Okay? Any questions? That's a good point. Oh, I see. Save. this. Okay, it should be there. Okay. Where has there it is. Sharing should be resumed. Oh, the lower sample? star sample. So you need to put something in the sample. That's so it's actually I just there was something else there just yeah, just copy this one file. So this is just working on one FASTQ file. So you may want to copy this file to your own scratch folder, or you can just work on the one that's in mine. I'm going to double check that.
I'm not sure this time should be.
Oh, has anyone not signed the timing sheet?
Ah.
So the sample equal is usually for this sample variable. So that in our sample, that's going to take. So whenever you see our sample resolution, same thing with this start. It's actually going to be replaced by this. Then this command is already valid one way. So if you just name out everything, you can. Just okay, this is where my sample has to be. This is where my directory is supposed to be. And then this is where the database is, and you just take full star and also change the genome index and its just this where it is. You know, same thing for each of the different star.
This performance, I was sure this last star.
Oh, one thing I didn't mention, but like whenever you have these Unix command lines, if you see a backslash at the end of the line, that means it's a continuity line. So if you type in the backslash in your main and then you return, it's actually going to not run the command right away. Go to the next line and let you type in the command. And it's actually just like a continuity line for the point. Yeah, it actually is. It's just really, and be careful because it's actually not going to ignore the line entirely. So you really need the space in this. This is weird because you're not supposed to have a star. I'm not sure why when you copy paste from the documents, and that's the same when I copy paste from the PowerPoint, but it's not taking in all the view lines. I can't do much about it. So if you just copy paste several lines, it's crazy and just detonate everything.
I was confused because I looked at the and I wasn't sure if this all captain star was.
No. Anything on the PowerPoint that's on two separate lines is really on two separate lines, except, I mean, it is reading on two separate lines, and the backslash means the command continues in the for the genome generator. So you need to be in the, it's just because where is your class? You're saying your class other places. Yeah, and I wasn't really necessarily expecting you to have done all this, but if you go to the workshop scratch folder.
Oh, yeah. You're
And now we have this GM directory. And so these are the assembly project object. And then this is the directory I was in from my friend. So one thing that's good is whenever you have a program, any program that's also contracting.com, just the dot slash from present directory. No, the dot slash literally means the address you're going to use from the present directory. And then it's directly into the second directory, right? So whenever you have a file that doesn't exist, it's very good to just but if you do your home directory, you don't need to. Oh, it's actually back to the genome index side. Yeah, so the genome index is starting in the so about the middle of the slide is start DB directory variable that's been defined to start DB year equal, and then there's a path into the cache folder of the workshop. That's where the start address is.
But when I was writing it, it said it's not compatible with the star version.
That's okay, it is not. Okay. So the most recent module on the cluster has been updated, and then they've also removed the older module. So we're going to use a different thing. We can't use the modules because the star database is, as it said, not compatible. So what we're going to do is, instead of using okay, so I'm currently in the workshop directory, and I've created this programs directory here. And there's a star executable here. So this is a star program itself, and this is what we're going to use. So you have two ways of using it. Either you type in the entire path to the program, like this. That's going to directly use a, so this is a help of star, so it worked. It started. Or you can do path equal this directory $path. This is the other way. And changing this variable here is going to tell the Linux or the shell that whenever you type star, you might be looking for something that's in this folder, and so it's going to find it because it is star. If I type star, now it's finding that file that we just showed. Okay. So if you type star or which star, it should show this path there. And if it does, then you're ready to go. If it shows you something else, you have to either call the file directly or tell the system where the file is. Okay. Oh, that's just to cd to the directory of the company, as in the yeah. So one dot always means the current, the present directory. Two dots, the directory of the present one. C1 dot works, but that does nothing. That changes the directory or the directory where you already are. Yeah. So that doesn't do anything, but it's legal. But it doesn't do anything. Right. So the programs directory is into the QC directory, and then I was in the programs directory. Then I was I typed cd dot, and then I was in the directory of other images. Oh,
that you can
sorry.
If you could comment again, because the only
Yes. I just did alt tab without really paying attention. Okay, so for the online students, what I meant is that if you go to the workshop directory, and then in this directory you will find this programs. So in the genome directory is where the star database is, and this is the one that's built with star 2.6, and the star module in the cluster is 2.7. And then if we go to the programs directory, this is where the star program is, and this is star 2.6. This one is star 2.6 inside the programs directory. And to run that particular version of star, that particular file, rather than the module one, you either have to call it like this, directly with the full file name and any option you like, for example, help, and that is star help. It's fairly long. I'll just. So this is just a star help, and so it works. We can call the program, and it is. I'm not sure if it's printing the. Can't see. No, it's not directly printing the version here, but it's 2.6. So this is one way to call that particular program. The other way is to update the path variable by just copying that directory and like that. And then even if you go to your home directory after this and then do type star, then it is going to be finding the star program that is in the QCB workshop programs directory. Okay, is this clear for the students?
Yes, they are. The slide for today, they are in shared Google folder.
The slides
In the shared Google folder. Yes, yes.
I need to refresh it. Oh, now. So there's an extra space somewhere in your command. Oh, one thing I can recommend is, especially with these commands that are long, just open a notepad document, like unsaved notepad document where you can just copy paste things and then edit the file as you go. And if you, I think you had the copy paste issue, so I'm not sure. But if you're just editing, so the backslashes are necessary on the slides because otherwise everything would be like all over the place. But I would not recommend using the back. If you're typing in the command, I would not recommend using backslashes. So it's great if you use backslashes. If you're directly typing the command, backslashes need to prevent the command from just when you arrow it's margin to come back fully at them. And then you're going to have to retype every line separately. Inefficient. Yes. No, that particular file that has trimmed in the name, it's already trimmed. If you want to go back to the city size, I've actually where did I put that? I showed the command, but I didn't give the full command. I can have a look, but I have downloaded Trinomatic in the programs directory. You have Trinomatic there, and I've written down the command so I could show it. Yeah, so if you see that, it's because it's not yet configured. So it is finding the right. Yeah, it's not finding the right one because it's finding 2.7, which is the one that's going to tell you that the database is not the right version. So let me update this slide actually. I'm just going to entirely update the PowerPoint slide. Is this wrong? Oh, come on, what's that? Okay, this should work here. And then when you do type star, then you should see this directory in the path that is returned. Questions? Actually, I think I can actually open the questions here as well. Chat. So this is this should be just place, but so this is like an entirely different command. It's actually I can't explain it. But spaces. So I think so if you want to check. You can always suggest to see what has changed. And actually, there is a start here, but it's actually basically the module is essentially what this is. So we include the module and then we put that again. Just copy paste one. Otherwise, just continue paste it from the log. Okay. Okay. So just you can't copy paste because everything is connected. And if you look at the lines, every line is one command line except for the one to slashes, which I mean if you try to do like slash, so right. So first of all, just so that we can just right. So it's entirely so seeing this first. And then it is okay. But it's not seeing the it's basically ignoring that command. So it's not like the path is twice like this thing there is what is actually seen by the program as an argument. So actually that is not an initial copy paste can't just do it like this. Slash like this. And that will genome here to this button, right? So if you have a dot in this variable,
Oh.
intro star.
Stable
This is your.
I don't know.
Yeah, it doesn't matter. It just became this way for some reason. You have to. Just so go back to star.
Not pretty much just do the same thing.
Cool.
And then type star. Just star.
So this is the dot, and this is in less, right? So just put this. So
Yeah.
it's running. I'm not sure which Yeah. So the help is not really telling sure which one. So this is not.
The beginning, you actually want us to
It's actually just
I don't know what.
visual freedom migrate. So you just can copy the so the program you have to use it or copy it, right? Because we're just using it as long as this is reading the program. You see the program. So it's fine. And then you can just do it. So what you want to do is there are only 5 words to read. So there's this P10 red one. Something that shows a sample we're working with here. And then we're having the sample variable defined as a people. And then the next line is a specific file, which is gas slash so that file just finishes in the
Yes. I don't know what I'm doing. I print all the signatures and then I look at the one, and then there's no file
that's where they didn't fully exist.
right? It's like
Yeah.
middle there. But am I like in the right?
I'm not sure what it is, but it's not working. Oh, I know what's going on. It's because it's not really handling the cheesy file the way it should. And then cheesy crashes, and the star keeps going even though cheesy crashes.
Okay.
And basically, whenever we see this error, okay. Now let me just can preview.
So,
Okay, sharing works. Okay, so I'm currently in my directory, and then there is this file that we're interested in, which is p10 rep1.trim.110. And don't type like line like this by hand. In general, you should be able to you should use type completion. .gz. Framed 1/10 dot. I think it's the right path. Let me just try. It's okay. It is the right path. So I have this path here. And then if I do try to do something with that file, like for instance, if I try to zcat the file, it's going to tell me something's wrong. And I can try like this. It's going to be the same, right? It's just telling me no such file directory, or it's going to be cannot find the file, or could not open the file, something like that. And whenever you see this type of error, and usually it's going to be like within a full command of a bioinformatic program and everything, and then something else might have gone wrong, and you can't be sure what's going on exactly. When you see this type of error, the first thing you want to do is go to the directory you think the program is running from. So in this case, I'm just in my home directory, and then so remove the colon if there is one, and then just use ls, the file. And ls is not going to lie to you regarding which file exists and where. And ls is just telling me, and so zcat in this case, I mean zcat, actually I'm using zcat, but then it's gzip that produces error. That's kind of what I meant the other day, zcat, gzip, they're the same program. Yeah, ls is telling me that there isn't such file directory. And it makes sense because this is what is in the current directory, and definitely that file is not here. So if that's where my program is running, and then even with ls I can't find the file I was looking for, then either I'm not running from the right directory, or there's a typo in the file name, or something. And then you know what's going on. You know it's maybe the path that has a typo in it, or maybe the directory the program is run from is not the right one, and then you can fix that. The other typical issue people run into is thinking the program runs from one particular place, but then actually the program runs from somewhere else. So you can just substitute ls to the program and then try to, it's basically reducing the problem where you have this gigantic bioinformatic command that crashes for some reason linked to a file, and then you replace all of that with ls to the potential source of the problem. Okay, just ls instead of the program. Does this answer the question a bit? So basically what you need is making sure the file is where you think it is, the name is typed without typos, and then the program is running from the place you think it is. So the program needs to be running from the right place, the name needs to be without errors, and then the file needs to actually exist in that place.
So the file not exists.
Oh, it's just like I'm looking for this file. I'm asking for this one particular file to be in the present directory, which is my home directory, right? And in my home directory, there is no such file. Like, because like there's only this to-do file and then these two other directories. But if I go to my Scratch directory and then to the QCB workshop, and then I do the same thing, I can just like go back in my commands and do this. Now it's finding it because that's where the file is. So I needed, in this case, I would have needed to change the directory. And same thing if I do zcat. Now it's also, it's also going to work. So, but generally the idea is that if less already does not work, then definitely your bigger bioinformatic program is not working to work any better. Just do it first time. Just do one at a time. Or I'm not sure. Permission. Yes. So anytime you see permission denied like this, like every time I've seen a permission denied, it was people are trying to create their own vectors into my spreadspace, because they're not allowed to do. Like depending, like on Unix, you have like multiple users, with each of them have their scratch base, and they can't cross visit their own scratch base. The only scratch space you can write in is your own scratch space. So you need to go to your own scratch space, copy the FASTQ file over there. The FASTQ file. This is the name has a dollar sample. So this dollar sample here is here in the the value of the sample variable. So when you ask how we do the programs can just see dot slash because entry is a dollar sample, it's not in the outside dollars. So actually the file is named. That's the file is actually named.
I see it, I just see what
It's not a page layer.
It's asking for more. They're not really
Increase it.
So that's with, first of all, you're on the login, so that's the immediate. I think if you're aware on the computer, you really want to use such. If you go back to the usage of Hoffman 2 slides.
So I need to exit.
No, I mean, you're already in the login, but you just once you use RSH -L and H RT. No, you want to RT. There's no so Control C. Yeah, so Control C just
I asked what's the same thing, but they said no, we have not tried. So automatically sends selecting.
that's because of the space. It's not seen by HTML. Control C.
Your treasure directory is already pushed back. So when you run this, they'll set it.
Magic.
So and you might even want to use 32 gigs if you want to open another terminal.
Right.
That might take longer than switch. So open a new terminal and then just just start. And then starting with this is what you're using for this terminal.
Yeah. So should I change it?
Just open a new one.
That's whatever we define.
And then the RSH was 32 gigs. If you Control C. And that one might works, then just use that one because it's definitely not going to keep you out. But otherwise, try to use a 69.
So I can
That
actually
starts some people, I've seen some people work with as long as you don't do anything. If you're running, there's a program that seems to get hundreds of processors. But, and sometimes you just ask for, like, I'm going to quickly talk this one word. Okay, but the actual point of the cluster is being able, the grid just has this scheduling system that puts you.
I'm not using it.
It's worse.
That's every time I read it down here. No, it's like
Resource allocation.
I'll show you.
Questions? If there is nothing the output directly, it is weird. Actually, I think it created the file from you might have
Yeah, so when you run.
I can't find the Zoom. It should take a few minutes to actually run.
Doesn't it?
No, that's not. I went into your thing. Now that's this part is do the recipe file again. Okay. And then copy the view of the just try to do that.
For some reason, it's not taking the output directory option.
You're alive on the hard
Okay. That's what. There is a very small, it's not, so nothing of this is preventing the program from running, and it's actually, so it's more of a, if this runs, instead of, where's my base? So here, when we define output directory, and then star actually does not see that this is a directory. It thinks it's like just a file name, and then it's going to use this, but it's just going to have a bunch of file names named like this with some extension in the directory it is run from. And so to fix that, which is was not the intended purpose, to fix that we need to add this slash here. And then star is going to actually use the directory we created. Okay, so let's work 5 more minutes on this. And if you can, if you see star running, then you're good.
So I just made that correction.
Yes, that's running. So it is running.
But previously it did update on this.
Yes. So instead of having this, you have this directory, okay? And then instead of putting this align sorted file in the directory, it just created like as if it just adds slash.
Okay. So should I continue? That's supposed to just stop me.
I mean, it worked. So it worked. And if you want, you can, if you downloaded that .sam file, and then you upload it in RGB, you get the same kind of results we had. Or you can just load samples. Oh, you were just working. So now you're going to have some files, but we don't have all the files yet. So these file things. So we have all these weird files.
That's part. No, I do the girl.
Very small. No, it's just
Oh, the cursor.
cursor.
Oh my gosh, it's incredible.
I don't know. It's you know, just trying to shoot it all. How long do you take to run? Anyway, this is your file for this if you just which is a tree that's that's simple, just this is one file.
You do that, you should just come getting it at the same time. Yeah, so you're telling Leslie that when you're allowed this, she should.
But what is it?
I did the pretty,
Yeah, that's the use of this.
Oh.
Because there's something going on. Then you did this should have been on a single day.
Such a big deal about it that you what it was.
But the thing is, you should just continue to manually trace it. Yeah, just always waiting.
No, what did I?
It should be the pre-screen when you.
Oh, this? Let me just make all my
Okay, any other questions before we move on?
Package code R something.
R?
R something. R something. That they use to do alignments.
Yeah, there are a few. I'm going to talk about it very quickly. So just to finish on these alignments, and I mean you're free to. I'm going to go through a few other slides just to show you what the other things I wanted to talk about today. Yeah, they can see the slides. Okay, great. And then you're free to just try again, work on the alignments. So the main things you want, there are a number, once you've done the alignments, once you've run the program, and the first things you want to check is whether the program has actually run. So you want to see if all the output files exist and if they're non-empty, and then if the error logs are free of errors. Like if somewhere in one of the logs the program tells you, oh, there was an error for something, for this or that, then it's pretty that you have to rerun the program in a different way after fixing whatever the error was. But even if the program runs, what you want to check is like the data itself. So, and the main thing that you will want to check is that your reads, reads created from this RNA library that you have prepared and sequenced. You want, for mouse and humans, you want typically more than 80% of the reads to align to the gene. That's the kind of numbers you should be looking at, okay? So you want to just take the BAM files, and then you can use SAM tools, which is going to tell you, for instance, the SAM tool flag stats that we mentioned yesterday. It's just going to tell you how many reads are aligned. And you compare that number to the original number of reads you had for the samples in the FASTQ file. And then if that's 20%, then something went wrong. Maybe I had a data set recently, it was human data, and then only 10% of the reads aligned to the human genome. Turned out there was some bacterial contamination for some reason after I kind of looked into it, and just 10% of the library was human. So maybe 10% aligned, or maybe 12%, and then 80% of that aligned to the gene. So if you don't see 80% of reads aligning, then there's an issue. That's really the one number you want to check. The other thing you can check fairly easily is that you see similar outcomes for the different samples you have, and that might be a way to see outliers. Or if you have two different libraries and you're processing everything at the same time, you may want to check that the two libraries overall have the same outcomes. If you have paired-end sequencing, you can look at reads that, I mean, every pair should the pair should really come from the same transcript, right? Because it's the same cDNA. And the two pairs should align in the same area of the genome, and STAR is going to tell you if that's the case, or so you can check whether or not it actually was the case and how often. If not, it's weird. And then if really you just go, things look relatively okay, and you just go ahead and then maybe try to do statistical analysis, but then you kind of see that some things are wrong, then you might want to dive deeper and do more quality checks. And there are some specific things, but it's not something I would recommend just doing by default every time. And you're always free to do it, but it's going to take time, of course. And yeah, the main one is really just checking the raw number of reads that align and the percentage of the original reads that this represents. Okay, so the second thing I wanted us to work on today was just once we have the alignments, and you can use the BAM files that are already aligned and that are in the workshop folder. And then we're just going to take this BAM file. So this is basically what our BAM file is, right? We have the reference genomes with genes, exons, and then we have reads aligned to those exons and genes with splicing. And then what we want is to get just counts for each gene, right? So this is the last step we're going to cover in the workshop. And we're going to do this with a HT-seq package. And we're going to count per gene counts. So that particular package can also do exons, but not transcripts. This way is good for differential gene expression. And the command in the package that we're going to use is called HT-seq count. So that's going to be the actual command. HT-seq is the package, or that's going to be in the publication, for instance. So we give it our alignments. We also give it the GTF file, so where the genes are, and then it's going to just look at all the exons and which genes they belong to, and then count how many reads it can find for each exon. There are like mainly three choices you have to make when you use it, this HT-seq count command. So the first one, you have to tell it you want to count to get gene counts, like per gene counts. So gene X is expressed at 200 counts, and the other one is expressed as 1,000 counts, 1,000 reads. The second one is you want to just let it know that you're looking for reads aligned to exons. And then the third one is going to be using mod union, which is good for differential expression. It's just a way it counts different subcases of reads overlapping things that are or not annotated, or potentially different multiple genes at the same time. Okay. And we're going to install it. It's very simple. Just copy this command. I can't install it for everyone, and there's no package, but it's very simple to install. So it's just going to take a minute. You first, on the cluster, you want to load the Python 3 module like this, because it's just not there by default on the cluster. So just load Python. And then we're going to just use the package installer of Python like this. You just type Python 3-m. This is a dash. This, I think, on the PowerPoint must be a converted hyphen, long hyphen thing. Like typographic hyphen is not going to work. The shell is not going to accept it. Just mentioning. Don't just copy paste the command, because I think that one thing is not going to work. and then pip install -- user. If you don't put the user, it's going to say some permission denied things because it's trying to install system-wide. And then you, of course, don't have permission to just change the system itself. And then the ht-seq package. And that should just install the software, right? So you have this in the PDF. Yeah, be careful about this hyphen there. That's not real hyphen. And then this is how we're going to run it. So basically what we need is a BAM file and a GTF file. So we're just going to use a BAM file for the P10 rep1 sample, which again, so that things run more quickly, I just remove some of the reads, just kept 1/10. Some of the alignments in that case. And then the GTF file is going to be the mus musculus GTF for the ensemble 98 version, which is in the genome directory in the Scratch folder for the workshop, right? Yes, so you have these two files. So this one is in the Scratch folder for the workshop. And then this is the command itself. So we just call ht-seq count. Is it? Yeah, so you will have just installed it. And then our libraries are stranded for this particular data. And then genes, exon in union mode. And then we want, the input format is BAM. And then we just give it the BAM file path and the GTF path. And then with this greater than sign, we just redirect the output, the standard output of the command into another file, which is going to be our output file, which we're going to call the same way as the input BAM file with an additional suffix dot per gene counts. Okay, so just this. Okay, so this is a command. I'm going to fix it right now. Remove that weird non-standard hyphen that Word has put there. Okay, this is. I mean, you can't. It's not in the PDF, but this is. Fixed.
In the next page, you have path. What does path stand for?
Oh, path to. This is meant so that. Oops. You're meant to replace this with the actual path to the file. So in this case, this is it's going to be a longer path. It's going to be flash scratch and Rochette, QCBW5A, genome. No, we didn't download that. I'm just going to show you what path it should be. That's the one. To switch to the right window. Is this active? Yes. So I'm here in the workshop.
Right.
And then there's this genome directory. And then GTF. Where do I have two GTF files? Oh, this is Jane code. We can remove that. These are basically the same information just from two sources. This is the one from Ensemble directly. This is also from Ensemble indirectly. Yeah, this is the one. So this is a actually path to this file.
Okay, I see. So instead of putting path, you should put the
Right, you just put this instead of path to, which is basically the QCB Scratch directory / enum.
Thanks.
Permission checking for path dot star exists and have permission to run. There. This is PWE. Just check where you are. Yeah, you're in my fresh place. Okay. So So what you want to do is this ASCII file. Once you you have to go to your own branch, so your own directory. You have to go to one of your own directories. If you're in non-directory, you're not able to. You're able to see the files, but you're not able to create them. No, this is everything is all in hot. But just so currently you're here, and what you need to do is to there, and then so you're going to replace so this is now you're in your directory. Oh, this is just you using. So now you were in this directory, and before you were in this directory, and then you go to version 5, and then so we're going to need to copy that.
You actually copy like this. Last time you mentioned this.
On Windows on Mac because terminal is better integrated with, and possibly if you Control Shift C, Control Shift P, okay, or just Google it. Yeah, okay. So there are ways to make it work. Okay, now most people who just don't have problems with Windows, but I don't exactly. And what we want to do for the start step is copy this. There's this file we use in this directory. Which is called, well, it's actually called. No, it's not this one. This is so for now, just for instance, I'm just trying to see if that's actually it's because so here, instead of $symbol, we use the value $symbol. Then it's actually this $P10.1 is actually at the computer flow. It was a name of variable, not content of the variable. That's just a content of the variable. So actually, because there is no variable that's named like this, the computer thought it was variable. I didn't translate. I mean, it's just property. Some languages will have this variable with this asterisk. Instead, it just replaces it with asterisk letters, as if it was an empty variable. Okay. So when you do this, then you don't have the actual. You don't have the actual. So if we move the $, and now if I do this, then it works. This is P10. Okay. I'm just checking that things are set up. And then the other way we could have done it was instead of just replacing the actual value on, because we have this $symbol variable that's already defined. So we're just changing the definition right now to the ASCII file. And it's still going to be the same because this symbol, the $symbol, that's going to be replaced by P. So this path is right. And what we do is copy the file. And currently we're in workshop file in your own fraction space. So we're just copying the other files here. You have to do this, and so you can run start from here.
Quick question.
You're back. So you need to do the path.
Right now. I'm not even sure if I will put you the same probably and run all the things again.
As long as you're not using, right, so the rest should be fine. Pretty much sure. Oh, this just types are doesn't really just shows you whether
It's still the.
so, yeah, it's not.
Yeah, that's.
And there's
Yes, that's pretty much right. They give you a general picture of last time,
install
first day, second day,
HTTP console.
just generate a basic and things like that. They have with that workshop too. So I saw that they refer to workshop too, just like value is almost like I didn't get it. So if you go back to workshop two, it makes so much sense. I think they also have the video online about workshop two that we have to watch. I think it's really awesome. The lecturers make clear concise. Yeah. They have the slide and also the video. You can actually watch it. Yeah. It's going up. Yeah, I saw the video. Yep.
So yes, I have not realized, but for the once you have installed PIP yourself, you still need to kind of fix something. It's just standard directory, but it's not actually using it. The easiest way to change it I don't have the Zoom window anymore. Oh, there it is. Yes, so this is what you want to use here. This tiled.local bin HTTP count. That's where when you install HTTP for yourself, that's where the program is going to be. Now this is a fairly normal place, but actually not by default. Yes. I think I just need to press resume. Yeah, do they see this? Do you see the mouse?
Not yet.
Resume share. Yes. I'm just going to do new share. Okay, like this should work, right? Okay, so I've added this part here before HTTP count. So yeah, this is where the program is going to be installed. Anything you install for yourself is going to be installed in this sort of directory, home.local. Yes. I can just try to make it work for everyone. I don't think it's going to work. Yeah, I don't think it's going to work. Just try to copy the program anywhere.
So previously I ran the alignment dimensions yeah. What does it?
Oh, well, it's ./.gz.gz. It's thinking this is a file name.
Okay.
And so it's looking for that file.
I see.
./.gz.gz.
Does it mean like it actually ran or not really?
No, it isn't. And it should exist, but star is not. Basically, star is using gzip to read your file. Then gzip is saying this file does not exist in caches. Okay, and star is still waiting for gzip to give it data without realizing that gzip has crashed. And really, star should realize that gzip has crashed, but it does not. So whenever you see errors in your log files, you should be concerned that something went wrong. Probably something good.
But actually take like 1/10 minutes and things that stuff.
I'm not sure what star is.
Imaging, and then they give me something like that.
And how big is this file? LSL. I'm not sure why it needed 10 minutes to realize that it didn't have any data to work on, but yeah, it just crashed. You need to fill the file name. So whichever you put in, so that file that gzip is trying to run is your FASTQ file.
Okay, here. So I should
So-
get rid of the dollar sign, I guess.
Yes.
Because you mentioned like if we are in the
So you can check the individual variables, like what is echo $ FASTQ file. It's going to show you exactly what you have put into the FASTQ file variable, and because this is what you're giving to. This is what you're giving to start, right?
Right.
So input file, the FASTQ file variable.
So I'm still a little bit confused about the dollar sign. I'm already in the present folder directory where the .gz file
It really
there.
is $ variable name.
Right.
So you're just writing a name. So
Right.
that's a name that's going to be replaced with content.
Okay.
So
So if
when
I
you do variable equal,
dollar sign
define the
variable,
content.
I'm defining a variable's name.
When you do variable equal.
I see.
When you do variable equal, you're creating the content.
Right.
And then when you're creating the content for a variable name.
Right.
And then when you do $name, you're replacing that name with content that is associated to it.
Okay. I see.
So
So
the FASTQ file is a
FASTQ
name.
file is a variable.
Yes. So a variable and a name is what.
Right. So in the future, if I'm trying to elicit the variable, I will put $FASTQ / file.
Yes, and that's going to be replaced with a command that uses it by content.
I see.
So and start just tries to open the file you're giving there.
Right.
And that's it.
It almost works, but they always say that this is regardless of where it exists. So
That's not the resource here actually.
So when I'm really drawing stuff,
What was the error on the directory?
this
Dot. Oh, so the doctrine, there should be a sample name before the trend. A sample, as in P10.
so it's not this.
It's not $P10, it's $3. If you go back to the PDF, $3.
Ultra sample. Okay.
It's actually $. Okay, so maybe if you want to, I've got like a couple more slides I could show just very quickly. And you're free to stay a bit longer, work on the exercises. So when you run RNA-seq, it's just running on one sample, right? You're just giving it one BAM file, so it's one sample. And then you have as many files as you have samples, with each the gene counts for the sample, right? And you need to merge all these files, and this is one way to do it. I'm not sure if you're going to use that in the R workshop next week, but that's one way anyway. There's like a million ways to do it. You can, whichever works for you is good. And then I had a section on running batch jobs on the cluster, which is going to be more difficult than running regular command, and it's just going to take time between every try you can make, and then this is just trial and error. I've put an example in the workshop directory. There's another type of batch job, which is really more complicated, which is also useful. I'm not going to talk about that. The last thing I wanted to mention was just like other methods, like more background regarding the workshop. There are a few aligners. So you might have heard about Top Hat. That's kind of the old aligners that were extremely widely used at some point, but they've been they are very slow, and they've been replaced by HiSat2 by the same research group. And maybe the aligners that's most used the most now is STAR, which we just are trying to use, which is widely used. Has very large memory requirements, which we kind of saw, and then even for creating the database, the requirements were even bigger. And then there is HiSat2, which you might also want to consider, which is good as well. They're just like different options. The aligner doesn't always make that much of a difference. Mostly, maybe the main thing is either it's going to really work or it's not going to work, depending on your libraries. Yeah, the aligner, it's the most difficult step to run in terms of computations, but it is not the most sensitive one. Like if it runs, if you have 80% or more of your read that align, then there isn't a lot you can do to improve things. Yeah, I just have this paper. I guess that's like maybe fairly advanced. The only thing you want to know is, depending on your type of data, you might want to consider whether your process is just the right one for the data. And then, yeah, the last two things are, we're using HT-seq. That's good for differential gene expressions. There's like a range of different approaches depending on really what your goals are for your analysis. So if you're looking at transcript, or if you're looking at just absolute expression values rather than at differential expression, you might want to use other things as possible. And then the last thing I wanted to mention was regarding this counts table, and that's kind of linked to next week's workshop. The counts table we're getting out of HT-seq count. So we're getting one column per HT-seq count run. This is not directly a table that tells you which gene is expressed more in which sample. And it's very obvious, but this table, of course, some columns, some samples are going to have more total counts than others, so you kind of need to normalize things. And this is where the CPMs, if you've been in the RNA-seq literature a bit, you've seen CPMs, and that's what this refers to, a per sample normalization with potentially some other tweaks. And then if you're looking at absolute values of expression between genes or between transcripts, then you also cannot directly compare one gene that has 2 exons with one gene that has 15 exons, because of course a gene with 15 exons is going to have more reads aligned to it. Okay? So there is transcripts per million normalization that's possible. Okay, and but for differential expression, we don't need to do that, and we don't want to do that. Okay, and that's the last things I wanted to mention regarding the counts table. And if you want more on the counts table, just yeah, come to the workshop next week. That's it. I'm going to be here. We still have 5 minutes of official time, I guess. So if you want to work, I'll be here. Yes.
Yes. Well, this was up here, so this was. Oh, that's. And so, but it's right, but
talk about some weird stuff. I highly recommend the workshop too, seriously. That one made so much sense. Yeah, like it just gives you a general idea picture about NGS. Yeah, this RNA-seq is NGS. They have a similar pipeline of course for RNA-seq doing the counting.
Oh, no.
Variation you care about.
So,
oh, I'm a medical student.
So then it's just because this one I think is actually.
No, I'm just.
So I don't think it produced so actually. Oh, this one's in. No, this is the one.
我现在是把这个 YP 的做这个研究的，我已经很 frustrated 了，我已经浪费了现在有人已经做了3次，就是我的做有一个手机的。
But it's very weird because it kind of lets you use the text, but at the time it's sort of has this standard combination, which makes sense to have a but it's not actually so no, it worked. It's just a matter of like sorting files out for different things. Yes. So power just means I'm just ignoring that you might need type to do this will go. So as long as you have a slash, the slash means just ignore the new line that's going to be right after. So if you type command, you can just ignore the slash.
Is this really funky?
Yeah, it can.
Yeah, it takes that long.
Well, this is just one pen for the, yeah, it should take a few minutes on the call. You take 7.
Okay.
Yeah, you should finish quickly.
Okay.
It's going to take longer on like the actual files are 10 times bigger. That's why you need the batch jobs, which we didn't go over that because it's just more time practicing with Unix. Yeah, and then you have 30 samples to process it 30 times. And it takes a few days, and then you just wait for the processor to give you station time.
For the test scripting study, so what's like the channel for sequencing downstream analysis? Sometimes he takes care of
I think, 50 seems very small to me, always.
depends which analysis of the data.
Yeah, it's
Sometimes it's for small library
Oh.
extraction, then if it's
Yeah.
for long, then you.
If you're looking at microRNAs or QARNAs,
Yeah.
which are getting to 20-ish bases
For
anyway,
messenger
then
RNA.
50 is good.
Yeah, I can see the transcript of RNA is different things. So some people are, the next will be not the back for the RNA analysis. So this one, we'll chop the sequence to pieces to align to the genome, right? So
No, I mean, like, even 150 is good, short read. So that's anything that's Illumina. Sequencing is short. Then Illumina typically has a short between 50, 100, 150.
yeah,
And 150 is pretty, I mean, it's pretty good work, but it's pretty more for like, transcriptome assembly, or if you, and it's not really for just quantifying transcripts in humans or mice. Right?
Yes.
Then 100.
Do you know which data you have? And which
I will have
those
yeah,
data?
we try with
And you
our
want to do like general analysis on the messenger RNA?
I mean, 50 base pair, if it's single end or single. I mean, if you're doing 100, I think it's really better. I'm not sure.
Yeah. And but you are interested in messenger RNAs, yes? So I think you can use longer. Because if you would analyze the short data, like microRNAs, so then
Yeah, if your RNAs that you're interested in are only going to be 25 bases anyway, then you definitely don't need 100 bases more, right? But if you're looking at the messenger RNAs, and actually for all the alignment and everything, having 50 bases is a bit short. Like,
and then you will have, like lot of reads, I think I don't remember exactly. I can double check because I did some kind of analysis. So then option on the messenger analysis, I implement. I think I put 100 or 100.
So you compare which lens will be better for.
Yes, it depends, like on the data.
but 50 would work. It just wouldn't be
Oh, yeah,
ideal.
yeah.
And then if you can just and then the thing is, it's not that much more expensive for the overall noise.
Cheaper, and I just want to count to compare the expression. That's it. It's a very straightforward.
No, but what I mean is that the entry analysis involving raising the animals and then all the characters and then all the data processing, that's going to take so much time. You kind of want to not make, I mean, that's kind of my, like, whenever you generate data, want to like make the data as best possible. So that, because if it's good but not ideal, you never know what might happen to just like there is a small issue and this small issue. Yeah.
Okay, thank you.
Should we each one? Are you around tomorrow?
Yes,
Should I just come to the dentistry?
Yes,
Like 10 a.m. or 11 a.m.?
I will let you know because I am with the baby at home now. She's sick. So
Oh, sorry to hear that.
yes, so I will like she's now with the nanny, so that's where I am. But I will let you know, so you know.
Yeah, okay. Yeah, just let me know if you are here tomorrow.
Yes.
Okay, thanks.
But it's okay. Bye bye.
It should be the one you just modified. Somehow I didn't take a picture of
I need to stop the recording. There's like so much work to do the Zoom thing. I mean, you pretty saw a bit of it, but no, it's not. It's actually recording the whatever I'm saying, and as I like switch slides, and it's just like this thing is